patterns of inheritance. In this chapter, by the end, you should be able to describe Gregor Mendel's experiments, the outcomes of a monohybrid cross, explain the relationship between a genotype and phenotype in an organism, and be able to calculate proportions in even percentages from a Punnett square. We will also be able to explain the laws that Gregor Mendel had instituted with his experiments, and this is the law of segregation and the law of independent assortment. And we're even going to look at how to identify certain inheritance patterns in non-Mendelian genes that don't follow a strict dominant recessive phenotype that you would see in an organism. And then we're going to look at how some genes are linked, what an epistatic type gene is, and some alternatives to dominant and recessive genes. So first, what we're going to do is talk about Gregor Mendel and his experiments. So a little bit about Gregor Mendel. He was fairly prevalent in the 1900s with his experiments. He did work as an Augustinian monk at an abbey in St. Thomas in Born. That is the Czech Republic. And he taught things to secondary and university levels like physics and botany and some natural sciences. And he decided in 1856 that he would do this long-term research project on inheritance patterns. And he decided to use the pea plant, this little guy right here, in order to do this experiment. And it turns out, if you go back and look at his research, his lab notebooks, which we still actually happen to have in a museum, and it turns out that he actually planted and recorded data over more than 30,000 different pea plants. So that's a lot of data. He did present and publish his data in the mid to late 1860s, and he published the data in this journal called The Proceedings of Natural History Society in a kind of article called Experiments in Plant Hybridization. And so you could go and find this copy if you wanted to in this particular journal, although I will say it's pretty dense and a little difficult to read. So what we're going to do is we're going to try to talk about what he did in a much more modern format. And so one of the things that we need to discuss is, why did Gregor Mendel use pea plants? Well, one thing, super easy to grow. And because of the number of seeds in a pod, each seed is considered an offspring, and that you can create tons of offspring from a single cross of parental organisms. You can also look at multiple characteristics, and we'll talk about some of those here in just a minute. And they're easy to pollinate. You can actually do it yourself. You don't need a pollinator, an insect pollinator or like that to be able to do this. So what he did was he actually followed several characteristics. We're probably only going to talk about a couple of those in this lecture, but seed shape, seed color, flower color, pod shape and pod color. And usually these were, either green or yellow, or tend to be the colors and stuff like that. Wrinkled and smooth. This, or round actually, is the phrase that is used in this image. And then you might look at the pod and say like inflated or constricted. So you could actually tell kind of the difference if you go and look at a pea pod, those characteristics yourself. So a lot of times the seed color and seed shape are the two most prevalently followed traits when we talk about these particular experiments in a science class, although we do tend to use flower color as well. And there may be a little bit more difficult characteristics to really see that he used, maybe flower position and stem height, although those are described using the same genetics as you would all the other characteristics. So let's look at the flower anatomy, because Gregor Mendel actually used the plants to do crosses, and well, in order to cross, you actually have to fertilize the flower to produce a pod that has offspring in it. And so the flower anatomy is very important. This section right here, the carpel that has the stigma, style, and ovary, is all the female reproductive organs. And you have the ovule here as well, which would be likened to the human egg in a female. And then you have the male reproductive organs here that are the anther and filament. And the anther is essentially what has all of your pollen. And to fertilize a flower, you would need pollen to land on the stigma, travel down the style, and then fertilize an ovule within the female reproductive organ. Flowers, obviously, there are multiple versions of flowers. There's that have different shapes to the stamen and carpel. And here's a little lily that has a variety of male and female organs. Here you have the female organ is a little kind of burst of multiple tentacles, if you will, and one here that has a much longer a style on the female reproductive organ. So just depending on the flower, the varieties of what those look like are very different. You also need to pollinate. So obviously what we would do is we would act like a pollinator, like a bee, in order to cross-pollinate a plant. And what Gregor Mendel would do is he would take a flower, he would remove all of the anther from a flower, and then he would use the pollen from a different flower and then place that pollen on the style or the stigma of the carpel and pollinate it using a paintbrush. And so you can actually do this at home if you wish to breed your own varieties or colors of plants, and then you can do that. Very easy to do. So before we get into the actual crossing and that kind of thing, you really need to pay very, very close attention to the vocabulary in this particular chapter. True breeding, hybridization, P, F1, and F2 are just some very basic terms that are used in reference to pollination and crossing organisms from to create offspring. And so true breeding is just a plant that happens to be a plant that can only give to its offspring one type of gene, period. And so we call it true. You might hear sometimes some scientists might use the word pure breeding instead of true. They both mean the same thing. Hybridization is the exact opposite of that, if you will, in the fact that a parent can give more than one type of gene to an offspring. And how did they get that way? Well, they had to have both of their parents must have been true breeding parents where they had different sets of genes that they could give. And so if I'm giving you an example, if you happen to have, say, a purple flower, which I'm just going to kind of draw over here, and then you had a blue flower like this, and I'm going to kind of draw these like this. Well, see, this plant can only give the blue flower gene, and this plant can only give the purple flower gene, right? So this is a true breeder, and this guy right here is a true breeder, but when you produce an offspring, depending on what gene is more prevalent in the sense of dominant versus recessive, I'm going to talk about that here in just a second, your offspring might look a particular way. So let's say this is the gene purple is the dominant gene. Well, the offspring, or the F1 generation, the parents up here would be the parent or P generation. This particular offspring may show that you only have purple as the flower, but it also has a gene that gives off a blue flower as well. And so if you were to take your purple flower here and cross it with one of its siblings that's also purple, right? Cross these two. Then it is possible in the F2 generation that you could have a combination of both purple flowers and offspring that have blue flowers. And my flower drawing is pretty bad. So this is the idea that Gregor Mendel wanted to show, and he wanted to be able to show some statistics about. He's very math oriented. And so he used true breeding parents, took data on F1 offspring, and generated F2 offspring, and was able to figure a bunch of stuff out that we know about today because of him. So let's see how he did this. So he took these P generation parents that were both true breeders. So they're using purple or violet flowers versus white flowers. Then his offspring in his F1 generation all had purple flowers. So that tells us violet is dominant. It's the dominant gene over the white gene. Okay. Then he crossed, see this little arrow right here? What he did is he took this plant and crossed it with itself, right? And then that plant produced offspring that we call the F2 generation. And the F2 generation, it turns out he did this where he counted and grew all of these seeds into plants and what color their flowers would be. Well, out of almost 1,000 plants, 705 were violet, 224 were white. And he was like, oh, okay, so this is some statistics that I can follow. And there's got to be a way that I can look at this mathematically in order to determine that certain traits are dominant versus recessive, and you can predict what your offspring will have at the end of that cross. So I've been using a couple of terms that we haven't discussed yet. So let's do that really quick. So some new vocabulary for you. Dominant and recessive. These two are very important that you understand that those particular vocabulary terms really are a make or break for you understanding what we're doing in this particular chapter. So a dominant trait, it's inherited into an offspring, and you will see what we call is the phenotype of that gene. So it's the observable trait that you see in an offspring, okay? A recessive trait is a trait that is not observable, okay? It's there, it's latent, or you might even hear the term carrier. So you might carry a trait but not have that phenotype. And so that's a really important term to understand, is that you can't see that particular trait whenever an organism has that and a dominant trait is there. That dominant will mask the recessive trait. An allele is a term that we use to refer to variations of the same gene. So for instance, if we knew that there was a gene for eye color, and we know that there's, brown eyes, blue eyes, green eyes, and of course there are multiple other colors. But these, say that these are only three. Well, these are all three alleles of eye color. They're variations of the same gene. Okay. Genotype. Genotype tells you what is the actual gene makeup that you happen to have in an organism, whether it's observable or not observable. And since most of the things that we'll be looking at are either pea plants or organisms like ourselves, those are diploid. And diploid organisms have two copies, or two alleles, if you will, of every gene. So if you happen to have the allele for brown eye from, say, your dad, and the blue eye allele from your mom, You've got two alleles, 1, 2. Which one are you going to have? Are you going to have blue eyes or brown eyes? Well, brown eyes is the dominant gene or allele, and so your eyes will be brown, but you're still carrying the blue eye allele, and you could potentially pass that on to your offspring. Now, the way that we typically talk about the genotype itself, we use letters to represent the dominant and recessive traits. Dominants use capital letters, recessive use lowercase letters, and we group two letters together depending on what dominant and recessive traits you have. So, for instance, if we're still using the brown eyes, blue eyes example, someone that was homozygous or has two identical alleles, if we use capital B for us, let's go back one more time. All right, capital B for brown and lowercase b for blue, then someone that's homozygous dominant, that means they have two of the same, two identical alleles and of the dominant. So they would be capital B, and that is their genotype. If they are homozygous recessive, that means they have two identical recessive alleles. So they'll be lowercase b, and that would be their genotype as well. What we call that genotype is homozygous recessive. All right. If you're heterozygous, this means you have two non-identical or two different alleles. You're going to have one dominant, one recessive in this case. So you'll have capital B, lowercase b, that's your heterozygous genotype. And we just call this heterozygous. You can say heterozygous dominant if you want, but that's a little, since it's heterozygous and there's really only one way for it to be heterozygous, it's a little overkill. Okay, so let's go to the next guy here. So let's look at, again, the dominant recessive, remember uppercase, lowercase. A gamete is the one of the alleles from your genotype. So if your genotype is homozygous dominant or homozygous recessive, then when you create an offspring, your genotype, you're going to give one or the other allele. That is your gamete. And this, for humans, is the egg or sperm. Okay, so this offspring, or this parent, say they're homozygous recessive, they can only give one lowercase y or one lowercase y, one each. Well, that's their only possible type of gamete that they can give. If an organism, what we do once you determine someone's genotype, you can then figure out their phenotype. And phenotype, remember, is the actual observed trait. Okay, the observed trait. So if you've got a dominant gene, then you're going to have that, whatever that dominant gene refers to in trait, you're going to have that phenotype. So capital Y refers to its dominant, so therefore it confers a yellow, say, seed here if we're talking about plants, and so that's the phenotype you'll see. Well, if you're homozygous recessive and you've got two lowercase y's, you don't have a dominant allele there. So therefore, there's nothing there to mask that recessive phenotype. So you'll see green seeds. Okay. So all of these there's a lot of vocabulary that goes into understanding this particular chapter. So it's really important for you to be able to do that. So what you want to do here is I want to give you a phrase, right? So if I say homozygous dominant, and you write for me the possible genotype that it could be, well, you know that that's going to be two capital letters. In this particular case, I'm just using Y's because I want to. Could you use the letter A? Yeah. Then you would write it capital A, capital A. Could I use the R, letter R? I could. Capital R, capital R. It doesn't really matter about the letter. It's just as long as you're keeping the capital letter as a feature. And then that phenotype for that organism that would be yellow because the capital or dominant allele confers a yellow phenotype. And that's something I would have to tell you what dominant and recessive traits are. If I said heterozygous, you would say that's capital Y, lowercase y, which would still be yellow because it has the dominant allele. And if I said homozygous recessive, you know, two recessive alleles, that's lowercase y, lowercase y. If I did this in the A's, I would make lowercase, lowercase, and the R's, lowercase, lowercase, so it'd look the same. And then you'd have that, whatever that recessive trait is, in this case, I said green. Okay, so let's try this. I want you guys to do this. So if I give you the parent genotype, I want you to tell me the gamete. So if it's capital Y, what is the possible gamete here? I'll take a pause if you need it. Okay, then you only have two options. It's a capital Y or another capital Y. If you're a parent that is heterozygous, capital Y or lowercase y. If you are heterozygous recessive, what are your two gametes? Lowercase or lowercase. It's very important that you be able to do this transition, being able to determine what the gamete is for the cross that you're going to do and figure out your offspring. So what happened is Gregor Mendel did all this math, right? He did all these analyses of different genotypes of different and phenotypes of different generations from P to F1 to F2. Well, there was another scientist, his name's Reginald Punnett, and his last name is spelt Punnett, that devised this really simple kind of square that was this, I call it like a graphic organizer or something like that, where you can quickly determine what the genotype and phenotype would be of possible offsprings two parents could give if they were crossed together. And then you could do some math about it, right? And so the Punnett square looks something like this. So if you cross a parent and a parent, these are true breeders, considering this is homozygous dominant and this is homozygous recessive, right? Homozygous dominant. And then this is homozygous recessive, okay? When you cross them, what you do is you make a square. And the square is what I call a 2x2. You have 2 rows, 2 columns. If you're only looking at one gene, 2x2. And what you'll do is you'll figure out what are the possible gametes from the first parent, and then you'll put that gamete, right, the same thing that's here and here, as a label for a column. So you'll put this Y here, down here, and then this Y here, down here. And then you do the same thing with the other parent, but do that on the row. And so it looks something like that. And so what would happen then? What you want to do is you want to take the label of each column and row and put them together. And that would create a genotype for a potential offspring. So you have a dominant allele, a recessive allele. So you have a dominant and recessive here. We call that heterozygous. And you do always write the dominant allele in front of the recessive allele. That's just kind of a convention that we do. And so now what you can do is you can look at, hey, there's a 25% chance, because it's 1 out of 4. This is 1, 2, 3, 4 possibilities here. We call that a 25% chance that your offspring in this particular box, this one right here, is going to be heterozygous. And it turns out that obviously if you go and look at the other genotypes in the other boxes, they're the same. So what we would say at this point is that all 100% of your offspring in the F1 generation, if this is considered your P generation, would be heterozygous. And that's your genotype. And the phenotype would be 100% of them are yellow, because they would all turn out to be yellow. So that's kind of how you use a Punnett square, and we do this of where we follow one particular kind of trait. In this case, we're doing something like seed color or something like that. And since we're following it in one trait, we call that a monohybrid because you're creating hybrids that are not true breeders. Monohybrid cross. So this is a Punnett square of a monohybrid cross. Okay. All right. So let's practice this. I'm going to give you, I want you to practice this on your own, really. I'm going to give you several different crosses. You got this first cross, second, and third cross here, and I want you to practice doing a Punnett square for these particular parental crosses. Now, I'm not giving you true breeders like what I did in my previous example, and that's okay. The actions that you take to determine the offspring genotypes and phenotypes and proportions or percentages, if you will, work in the exact same method. So once you try those on your own, you can pause this if you want to. And so when you get done, we'll be ready to go. All right. So the second thing that I want to show you is how to do a Punnett square of what we call a dihybrid cross. So this dihybrid means you're looking at two gene types at the same time rather than just one. This is going to require that you have two sets of letters that refer to each one has a dominant and a recessive. So what I did is I went ahead and assigned them the letter, the dominant and recessive uppercase and lowercase, and then the trait, the phenotypic trait that is associated with those, and that's over here. So this is kind of what I call like a little key for your gene to trait organism cross. So capital A, the dominant out of the flower color category. This would be flower color. The capital A refers to purple flowers. The lowercase a refers to white flowers. And then the second type of gene is going to be seed color. Capital B would be yellow and lowercase b would be green. Now, before we've been using Y's for yellow and green. It doesn't matter. It doesn't matter. We can pick whatever letter we want, just as long as you can tell the difference between the capital and lowercase. Okay, so before we actually do a cross, where we're going to use a Punnett square, what you need to have is the skill in order to determine what the genotype of the gamete will be. This one's tricky. It's a little tricky in the sense that the A's, the gene for A and the genes for B are independent of one another. So whether you get one dominant or recessive gene of letter A or dominant or recessive of gene B, it doesn't matter on the other. They have no kind of influence on one another. So essentially what you have to do is write potential gametes with a single A and a single B together. Because you're going to give, it's kind of like saying, hey, your parents have a particular eye color and hair color. Well, they're not only going to just pick one, you're going to get a trait from your mom of both eye and hair color at the same time. And you're going to get the same thing from your dad. You're going to get an eye color and hair color from your dad. So we're just doing that same thing. So to determine the gamete for a parent that is homozygous dominant for both traits, this means flower color and seed color, capital A, capital A, capital B, capital B. Each gamete possibility will be homozygous, will be dominant for those traits. And why am I writing this 4 times? Well, it turns out, and let me write this down here just so you can see. There's that parent's genotype. This A, I'm going to call it A1, is a different allele than that A. They're actually on different chromosomes. Okay, they're just on the same location. You got this one, this person, this particular plant here, got this A from its mother and got this A from its father. Same thing here, from its mother, from its father. So they're actually all on different chromosomes. So if I labeled them 1 and 2, 1 being mom, 2 being dad, what this really looks like is these guys over here are A1B1, A1B2, A2B1, A2B2. So you're actually, because these are on different chromosomes, you're indicating that with the gamete possibilities. Okay, so it's very important to be able to do this. A lot of times how I actually figure out how to do this, I use what I call the foil method. The foil method, where I use the first two genes. This would be 1 and 1. I'll use the outside 1 here, inside here, here, and then last 2 and 2. So let's try that foil method with this second parent and see if we can figure it out. So it's A, lowercase a, capital B, capital B. So we're going to use the first two genes, capital A, capital B. That will be one of your gametes. Then you're going to use the outermost. That will be this A and this B. Capital A, capital B. Okay. Then you're going to use the innermost, inside, lowercase a, lowercase, or capital B. Yeah. And then your two last, lowercase b, capital B. Or lowercase a, sorry, capital B. This is how you go about doing the FOIL method. So why don't you try doing the FOIL method for this one and see if you can follow the same pattern. Okay? When you get that done, or pause it if you want to. When you get that done, we can come right back. Okay, so hopefully you figured that out. Let's write our answers here and see if they match. All right, so those should be your genotypes. If you use first two, first and last, two inside, and then last and last. All right. Okay, so now what we're going to do is, what you've realized now that you're following two sets of genes, you get 4 potential gametes each time you try to figure out the gamete. Genotypes from a parent that has 2 sets of genes. That means you're going to need a Punnett square that's a 4 by 4, which you see down here. So let's see, I kind of wrote under there. Let's try doing this with this particular cross right here. These are considered true breeding because one parent is homozygous dominant for every trait and one parent is homozygous recessive for every trait. So these would still be true breeders. Okay, so let's take our first parent here and let's figure out what its gametes are. So we're going to write our first, second, third, and fourth gamete. And I realize it seems like I'm going really fast through this. I've done this a lot of times, so I'm a little bit kind of a pro on it. But if you need a little bit more time, then just pause the video or rewind so that you can try and practice it before I write the answer down. Same thing for your other parent. All of its gametes are going to be lowercase recessive genes. Okay, how do you put them together in a single box that represents a single type of genotype for an organism? Well, what I do is I typically take the first 2 letters, or the A's, in both of these gametes, and I put them together. Capital A, lowercase a, and when you're writing the genotype, the full genotype of an offspring or organism, write the dominant first. Then you go to the second letter and then write that second letter with a dominant first. So you have a capital A, lowercase a, capital B, lowercase b. It follows the same look and feel of the parents, where the A's are in front and the B's are in the back. Okay, so if you did this for every single offspring in this particular Punnett square, you would realize every single one of them, all the way down, all the way across, so forth and so on, are all going to be this same genotype. Okay? So, you know 100% of your offspring are going to be heterozygous for every type of gene. This is the flower color and the seed color. And you can even figure out what color flower and what color seed this offspring will have. So it turns out that you will have purple flowers because you have a capital A. Even though there's a white allele there, a white gene allele, there's a dominant allele there, so the white gets masked. It's not present. And then it turns out you will have yellow seeds. Why? Because you have a yellow seed allele, even though there is a green one. Okay? Every single one of those offspring will be that. Okay? All right. So I kind of go through a little bit underneath there. Alrighty. So let's let you try this with another set of parents, if you will. We're going to take the same genotype you generated in the F1 cross of the previous slide. So these are going to be heterozygous organisms, and you're going to determine how many offspring there are going to be with their particular genotypes. And then we're going to count them and do some statistics. So pause it if you need to. I want you to generate the gametes that go in the columns and the rows, and then put together the offspring genotypes, and then we're going to count everything. Okay, well, so hopefully what you got for the parental column gametes are capital A, capital B, capital A, lowercase b, lowercase a, capital B, and lowercase a and lowercase b. And it should be the exact same for the other parent here for over there. You put these together, you'll have all of these offspring. You might need to pause the video just so you can take a gander at all the differences in phenotypes and whatnot, or genotypes. But if you don't want to, let's just keep going, and let's talk about what kind of thing you're going to see in the actual offspring phenotypically. So are you going to see purple or white flowers? Are you going to see yellow or green seeds? Well, it turns out that if you go and look at the proportions, so if you count that there's a total of 16 possible genotypes, the proportions are kind of crazy in the fact that, you know, you've got like 1 or 2 or 4 versions of the same kind of thing, and they all add up to 16. This would be 16 out of 16. This is not super useful, the proportions of genotypes in a dihybrid F2 cross. This would be like your F2, because you used the F1 parent offspring to make this cross. What's really important is the phenotype, and this is how many purple flower yellow seeds, how many purple flower green seeds, and so forth and so on. And if you're always starting with a parent, a P generation that's true breeder, and then your F1 is going to be heterozygous for every trait. So it'll be something kind of like this in their genotype. Then in your F2 generation, all right, you will always get this proportion of what we call 9 to 3 to 3 to 1. That is always the genotypes you will get, the proportions of those. So let me show you the purple yellow. These guys right here, I'm going to kind of mark in a purple over here. So this guy will be purple yellow, purple yellow, purple yellow. So all the way through. All of these. And it turns out that there's a total of 9 of those. Then what we're going to do is we're going to do the purple green. All right. This is purple green, purple green, purple green. All right. And then what we're going to do is this little red one, yellow, white, yellow, white, yellow, white, yellow, white, yellow. And then white, green, 1. And if you do your Punnett square correctly in this format, this is exactly what it'll look every time. All right, so let's move on to those laws that I kind of talked to you about a little earlier. We've been using these laws, you just didn't realize it. There's 2 laws that Gregor Mendel really was able to tease out for us, although he probably didn't use these terms as laws of something. We just used his data in order to present this in a more cohesive way. And the 2 things that he came up was this thing called law of segregation, where what we call paired factors, these are genes. So like if I had capital A and capital A, these are some paired factors or paired genes, must segregate equally into a gamete. This means you have a 50-50 chance, 50% chance of getting either this A or this A, meaning whatever gene that's referring to. And that's what we were doing with the monohybrid cross, where you're figuring out from one gene, you have one gamete versus another gamete, and then you kind of cross those together. The second one is the law of independent assortment, and this is where genes do not influence each other with regard to this law of segregation or sorting. So this makes where every combination of alleles exist, and that's where you'll see in your dihybrid cross like this. So say I had this genotype, and I know that the genes that A's refer to and the genes that B's refer to have no bearing on one another. So if I pass on capital A or lowercase a to an offspring, that will not dictate whether they get capital B or lowercase b. So this is why we use the FOIL method in order to determine what are all the possible combinations of both of these sets of genes. And this is where you'll get these gametes as options, because neither one of them influence the other. So these are the 2 laws. That's how we refer to them in a definition sense. Okay, so there are several genes that do not follow the Mendelian inheritance, and these are called incomplete dominance, codominance, sex-linked, and epistasis. So what I'm going to do is just show you an example of each one of these so you can kind of sort of identify how they work. All right, so incomplete dominance. The way that incomplete dominance works is it looks incredibly similar like what you have seen before, where you have a dominant allele and a recessive allele. You will have a dominant allele, which you can see in the genotype of this red flower. Oops, sorry, wrong way. And then you'll have a recessive allele, and you can see that in the genotype of the white flower, or the phenotype of the white flower. The problem is whenever you get an organism that has one dominant and one recessive, the outcome of the phenotype, all right, no longer follows dominants versus recessives traditionally. Instead, what it does is it blends the 2 traits. So you would get, the classic capital R, lowercase r for the genotype, but see the phenotype won't be red flower, it'll be a pink one, because it's a blending of the 2 traits. And so that's what we just call incomplete dominance. Now, how do you know by looking at the genotype of an organism, say 2 parents? Say if I said, hey, you have this parent, cross it to this parent, how do you know that the offspring are going to be pink if they get heterozygous genotype? You won't unless I said, hey, these 2 parents cross and their genes, their phenotype is determined on incomplete dominance. So I actually have to use that phrase in order for you to know that when you create your offspring, which you're going to have a set. So if we did our Punnett square that looks like this, like that, then you'll have half your offspring will be homozygous dominant genotype looking. They'll be red. These will all be red. But see, all these are heterozygous, so they won't be red, but because they're incomplete dominant, they'll be pink. And so it's just the phenotype that looks a little different. The Punnett square and everything looks the same. Let's try that one more time. You're going to take a cross of a heterozygous flower. What color phenotypically is that plant going to have? Pink. Pink. So we figure out the offspring. You're going to have one of each allele. Place those on your rows and columns. Then you're going to place your offspring genotypes. What color is this offspring going to be now? You had 2 pink parents. What's this guy going to be? Well, it turns out it's going to be red. Then these guys right here will be pink. This will be white. So this is kind of the kind of a further down or another example of what the incomplete dominance might look like. You can do some proportions of genotypes versus phenotypes, if you will. Really easy to mathematically determine those. Okay, so another version of non-Mendelian here is that second one is called codominance. And codominance means you have 2 dominant alleles. So you're going to have 2 alleles, 2 capital letters. And in this case, we will always use different letters for the dominance. So in this particular case, the alleles are going to be capital A and capital B. Both of them are dominant. And where this is really like a good example to look at is blood type. And it turns out that humans, and blood type in humans, if you have one or both or none, you could potentially have none of these alleles. Your blood type will be dictated based on what kind of protein is built on the outside of your red blood cells. So if you have the gene for A, the dominant A gene, then you're going to grow and put these little specific looking proteins on the outside of your red blood cells. And they're in this particular drawing, they're like look like little lollipops or spheres on the end of a little stick. If you have the B gene, then you're going to have these little kind of what looks like in this drawing, little kind of triangles or diamonds. Which you'll see right here. If you happen to get both of these alleles, they're both. You're going to see them both. That's where the codominance. You'll see both of those traits. And so you'll see the little circle and the little diamond here in someone that has both of those genes. If you have neither A or B, you will have the little kind of stick that's there, but you will have no actual circle or diamond in this figure that you see, or the protein that corresponds to A and B. Okay, so you can actually follow a parent's genotype for their blood type and what the possibility of blood types are for an offspring. Now, keep in mind, remember, every person has 2 copies of a type of gene. So the type of gene we're talking about here is this little protein that goes on a blood cell, red blood cell. Now, a little weirdly, the alternative option is no gene. But say if you're group A, you have blood type A, you could potentially have 2 copies of A. This gives you blood type A. Okay, but you could also have 1 copy and no copy for that second one. That's still blood type A. So there's 2 versions, 2 genotypes that can give you that blood type A. So what would blood type B look like? Capital B, 2, or only 1, B and O. O meaning the absence of a gene. Not that it is another gene, it's just the absence of. This is still blood type B, blood type B. There's only really 1 option for AB blood type, and you have 1 of each. And then for O, you just have O. Okay? All right. So if I give you a parent's genotype on blood type, Can you determine what the offspring look like? Yeah, we can do that. So let's try this. Now, I will say that whenever we tend to do Punnett squares using blood type, we tend to write it a little weird. We like to have, with the presence of a gene, we put I, the absence of one, we put lowercase I. And so if there's a capital A, we put it as a subscript, capital B, subscript, and then the lowercase I, it's, you don't have that particular allele. So a little bit weird. Do you have to do it with the I's? No. You could just do it with the zeros and take place of the absence of a gene. So it's just kind of, what's your preference to do. I will say this is kind of sort of convention, but okay. So let's say if we take a parent that is A blood type and a parent that is, this would be B blood type. What are the possible blood types their offspring could have? Well, you treat it just like a regular Punnett square. 2 by 2 is all you need. And what you'll do is you'll go, hey, this is one allele and one allele. Those are going to be both my gametes for that parent. And then this is one allele and this is one allele, both of them gametes for that parent. And then what you do is you cross them. You put them together in the square. So this person right here is AB blood type. AB. This is A and nothing, so that just says A blood type. A and nothing, that's A blood type. So is it possible for a parent, right? So this is the kind of question I might ask you. Is it possible for you to have a homozygous dominant A blood type parent to a B blood type parent to have an offspring that has B blood type? Answer to that, no. Okay, so this is a no. So maybe take a moment, see if you can do this kind of problem on your own, and then we'll go from there. The third type of non-Mendelian inheritance is called sex-linked, and sex-linked means just that the particular allele you're interested in, the gene you're interested in, happens to be on the X or Y chromosome. That's it. So you can actually associate it with male or female as well. So I can say, hey, how many females are going to be brown-eyed, or how many males are going to have blonde hair, that kind of thing. More often than not, we tend to show examples of fruit flies, because fruit flies was one of the very first organisms ever really studied when it comes to sex-linked things. And the kind of major guy was Thomas Hunt Morgan in the early 1900s. Oh, that's Morgan, Thomas Hunt Morgan. In the early 1900s, published some studies on sex-linked genes and how they pass from parent to offspring. So this is just giving you an example of what a female fruit fly and a male fruit fly look like. Obviously, females will have 2 X chromosomes. A male will have 1 X, 1 Y chromosome. And this is just the traditional or atypical genes or chromosomes you would have for male and female. Okay, so this is kind of how it works. We typically use the X as indicating that a particular allele is on the X chromosome, and the Y saying that it's on the Y chromosome. This is why you'll see X and Y, and then a superscript as telling you what the gene of interest would be and what the letter. So this capital W represents what we would just have used for capital A's and B's and R's and Y's and whatever that we had done previously in normal Mendelian inheritance. So what will happen here is let me show you how this works. What we're following here is eye color in the fruit fly of males and females. And the 2 colors are red and white. Red being capital W, white being lowercase w. Now, I will say that sometimes this is difficult to see the difference between capital and lowercase when the letters look very similar to one another. I don't like using letters where they're really similar, but here we are. You can kind of sort of tell that these are lowercase compared to this one up here that's uppercase. So, this is something we're going to have to be aware of. So, the male that you see here has one X, one Y. The X has the gene for eye color, and the color is red, so therefore the fly should have red eyes. This female down here has two X chromosomes, both lowercase, lowercase. That means recessive allele, lowercase. Two of them, that means it does not have a dominant allele there. It's going to have that phenotype of white. So these guys are red. Male is the red. Female is the white eye. Okay. So if we do that and we put them in the Punnett square, almost always do we put the males over here on the side as the rows and the females on the columns. And what you'll do is you'll kind of take those and put those together and you'll get a phenotype. So you'll have red, white, and this is female. So I know that this is female. It has a red gene and a white gene. It's going to have red eyes. That's how that works. You can tell female and the phenotype at the same time. And so this is kind of how that particular type of cross works. This is just how we tell if something is sex-linked. All right. The other kind of non-Mendelian inheritance is called epistasis, and epistasis is a little more complicated. I'm not going to have you guys doing Punnett squares of epistatic genes. I just want you to kind of understand the concept behind it. And the concept is saying that if you have the expression of one gene to be a certain thing, then it means that that can affect the expression of another set of genes. A lot of times coat color is a major player in epistatic genes. So you might have the presence of C. If you'll notice here, there's capital A, lowercase a, capital C, lowercase c. And it turns out that for the coat color, what you'll notice, and you're trying to figure out what's epistatic to the other thing, what is the one that causes the other one to be affected? And it turns out that if you look at these guys, the little white mice, see how they all have lowercase c? Then they don't have a gene that can say, hey, you need a coat color. All right. So the C, the gene C, is epistatic to A. And the A is the coat color. And that, the capital A and lowercase a, are what dictates you having a particular color. And how do you know which one is which? Well, all of the mice that are, let's see if I can figure out, all of them that have brown coat color, if you will notice, all have at least one dominant. So dominant must be brown. All of the ones that are these darker coat color have no dominant A allele. So that must mean that these are black. And that's how that works. This is kind of the idea behind epistasis is one gene, one set of genes influences another one. Multiple situations where that happens. You can have the epistasis works in the colors of your pepper that that's green, yellow, and orange peppers, the large bell peppers. Even in coat color, say for the lab, the yellow, chocolate, and black labs, that would be an epistatic situation. So again, you guys are not going to have to do crosses of them. You just need to really understand the process of epistasis, meaning one gene influences the outcome of another. Okay, so hopefully in this video you have learned how to describe a little bit of Gregor Mendel's experiments, the outcomes of a monohybrid cross, the differences between what the term genotype and phenotype mean, how to do and calculate proportions or percentages, if you will, in a Punnett square. Be able to tell me the difference between segregation and independent assortment in the reference to Mendel's laws. Give me a little bit of information on patterns of non-Mendelian inheritance and what kind of things in those non-Mendelian inheritance are important, like gene linkage, meaning, if you have genes linked to a particular sex linked or, in that case, epistasis for genes influencing others. And just as alternatives to dominant and recessive would be that codominance or incomplete dominance. Okay, so what you need to do now, make sure you read the chapter, complete your learning guide, create your own set of chapter notes. It's really important that you take some time to make your own notes. This is a part of your studying. You want to create any additional material. You might want to make some flashcards. You could probably use Quizlet if you want to instead. Maybe make some diagrams. If you want to make a sheet of paper and put it in a protector and put a little Punnett square in there, and then you just practice it over and over and just erase it. There's some additional video content on YouTube about Punnett squares. I highly recommend you go watch those. And then it might not be a bad idea to get a study partner to help you learn and teach each other of these concepts. All right,